Good morning, everyone. Welcome to the webinar. We're going to wait to start at 8:03 once the guests have trickled in. Okay, we're going to get started. Welcome to the webinar from the Research Coordination Network funded by NSF for SARS-CoV-2 research on wastewater for wastewater-based epidemiology. This webinar is for junior researchers. Those are researchers who are at the early stages of their careers, postdoc students, assistant professors. Thank you so much to the two speakers who are joining us today. We have two speakers, Maitreyi Nagarkar from US EPA, a postdoc, and then Devrim Kaya, a faculty research assistant professor at Oregon State. Our first speaker is Maitreyi. So Maitreyi, can you go ahead and share your screen? While she's sharing her screen, I'm gonna explain to you all how the webinar format works. Please put questions that you have for the speaker into the Q&A. You have the ability to upvote questions that you would like to hear the answer to. Do not put questions in the chat because we don't monitor the chat for questions. And at the end of the 20-minute presentation, a moderator from the panel here will ask questions in the Q&A of the speaker for about 10 minutes. So please start putting questions in the Q&A as they come to you, and especially towards the end of the webinar presentation. With that, I'm going to pass off to Maitreyi, who's going to tell us about her work. Thank you.
Thank you. And can you see my slides and hear me okay?
That looks great.
Yep.
Okay, perfect. Thanks. So yeah, thanks for the introduction. As you said, my name is Maitreyi, and I'm a postdoc at the US EPA. Our group is based in Cincinnati, Ohio. And so I'm going to talk a little bit today about our work doing, long-term quantitative monitoring of wastewater for SARS-CoV-2. We started almost a year ago. I think some of our first measurements were at the beginning of May. And then I also wanted to share some information about our preliminary sequencing efforts. And firstly, I would, of course, like to thank a very large group of people that have contributed to this work. So at the US EPA, particularly, I want to thank Nicole Brinkman, who kind of coordinates all of this, Chloe and Emily do a lot of the lab work. Scott and Mike have contributed to the modeling, and Eunice and Jay have been, on a higher level doing coordination work. We also have a number of other contractors that work in the lab, and then we collaborate with the state of Ohio and several different universities in Ohio as part of this broader monitoring network. And of course, we'd like to thank the utilities that provide us with the wastewater samples. So to begin, I don't think I need to explain too much about wastewater monitoring to this particular audience, but as we all know, SARS-CoV-2 is shed in the feces of some, although not all, infected patients. And the way that I like to think about these things when we're thinking about what a wastewater sample represents is in the schematic that I have here. My horizontal blue lines could represent the duration of fecal shedding for any given individual. And then the red squares represent a positive test over time. So when we're taking a wastewater sample, what we're representing in my highlighted slice is all of the individuals that are shedding at that time. Some of who have gotten tested, some who haven't, some who never will. But what gets reported as case data is just those 2 positive tests from that day, right? And so a lot of the wastewater monitoring efforts are trying to connect one type of measurement to the other type of measurement. And of course, we know that viral RNA can persist for at least a few days in wastewater. And as this webinar can attest to, the surveillance is happening worldwide at a number of different scales. And I think the sequencing efforts are starting to become more common. The project objectives, so we do consistent and high frequency monitoring of SARS-CoV-2 RNA in wastewater. At the US EPA, we're focusing on 12 sites in Southwest Ohio in conjunction with this broader Ohio statewide network. And so we report that data on a weekly basis, and the state is using it for rapid public health response. We also have been working on some methods optimization on our end, partially because different groups in the network use different methods, and so there's also regular interlaboratory comparisons that occur. And then finally, there's a major goal of modeling the relationship between what we're detecting and cases, and a big part of that is looking at normalization factors, or things that can be incorporated to the model, like fecal loading in the wastewater, sewer shed size, and so on. And then we want to do sequencing to start to detect, hopefully, mutations that are associated with the variants of concern that we see emerging in clinical samples. To briefly touch on our processing methods, because a lot of different groups process different components of wastewater, we receive 24-hour composite samples, and we spike them upon receipt with OC43, which is a human coronavirus, so that we can use that as a process control. We take a small volume direct sample from that, and then we spin it down, and we have a pellet fraction. We filter the supernatant for a filter fraction, and for a while early on, we were also doing ultrafiltration. Once we do all of these, we treat each fraction separately, and so we do DNA and RNA extraction, and then subsequently quantify using droplet digital PCR. We are looking at N1 and N2 targets for measuring SARS-CoV-2 concentrations. We're also, like I said, we spike OC43, and so we can have those measurements to assess recovery from our extraction and other processing. And then we look at inhibition, and we're trying to explore fecal content using some fecal indicator targets, including PMMOV, crossphage, and HF183. Our turnaround time for this is about 3 days. So because we are actively reporting to the state of Ohio, I'll talk in a second about why we ended up eliminating the ultrafiltration step. But so what we report is the pellet and filter concentrations together as that wastewater concentration. And just a few logistical, things we found. We didn't find a significant difference in N1 or N2 measurements when the samples were held for up to 4 days at 4 degrees Celsius, although we did see a reduction when there was a freeze-thaw cycle. So, and then finally, we are now beginning to sequence from these RNA extracts as well. Really quick, just again, one method consideration. So like I said, we eliminated the US step because here what I'm showing is 3 different targets, cross-stage, PMMOV, and then N1 and N2 for SARS, at 3 different sites, Lick Run, Mill Creek, and Taylor Creek. And we're looking at the concentration in copies per extract in these different sample fractions. And so what we found was that over 90% of the nucleic acid were always found in the pellet or filter fraction very consistently. And so because of that, and because of supply chain issues, we discontinued use of the UF. And so we're looking basically at the solids in the wastewater. Once we do all this, we report that data, like I said, to the Ohio Water Monitoring Network. And so the state of Ohio, in addition to kind of the standard dashboard that has hospitalizations and cases and so on, has a tab for the wastewater, like a wastewater dashboard. And so there's about 65 sites, I believe, that cover, you know, in their combined sewer sheds cover about half the population of the state of Ohio. And so if you go to that dashboard, what you see is this map with all the different sites. There are some, you know, a summary of kind of the general trend. So whether at that site from the previous week, it's been increasing, steady, or decreasing. The screenshots from a couple of weeks ago, I think, but this is kind of what it looks like. And then if you click on an individual site, so we, like I said, Cincinnati, we're in the southwest corner of Ohio in Hamilton County. And so Mill Creek here, that's the largest sewer shed within our county. What you see, again, screenshots from the website, is a report of million gene copies per day. So that's flow normalized to look at how much is flowing through in a day. And then you can also look at, in both of those cases, it is N2 copies. And so in the bottom, it's copies per liter as the bars, and then the total number of cases as a 7-day moving average in terms of, you know, folks that tested positive in black. So you can look at that for every site. And we have been seeing at our sites a pretty good correlation between our wastewater measurements and the case numbers. And so just, you know, the objectives of this network are, you know, to try to serve as an early warning of infection, and then therefore provide information that might inform community interventions. And because, you know, we have this high frequency time series, perhaps also assessing the effectiveness of those interventions. There's a number of efforts within the state to develop predictive models to translate our wastewater data and how it compares to other types of data, like community infections measured with clinical testing and so on. And then, of course, we can look at disproportionately affected communities or communities that might have fewer resources for testing as well. On the research side within US EPA, some of the questions that we're looking at are, you know, sampling at different spatial resolutions. So for example, we look at Mill Creek, which I mentioned is the largest sewer shed within Hamilton County, but we can also look at Lick Run, which is a more vulnerable, impacted sub-sewer shed. And what we found is that the cases, you know, the increase in wastewater concentration, this is copies per liter of N2, in Lick Run has tended to precede that of Mill Creek. And so we wonder if certain sites like that could serve as early warnings or sentinel sites for a broader community. And then of course we can compare different types of sewer sheds. Mill Creek has much greater flow. It has industrial inputs and combined sewers, whereas this adjacent sewer shed has lower flow, serves a smaller population. And so we're hoping to look at how sewer type can affect some of our measurements, including how we can best measure fecal input. So a number of groups use cross-stage or PMMOV, which are fecal indicator targets. And then we also know about flow. So for example, Mill Creek has has about 40% combined sewers. And so we might suspect that rain events that lead to increased flow might dilute the fecal signal. So we're working on trying to incorporate a number of pieces of information into models that rain issue might not be as big of an issue in something like Taylor Creek where there are not combined sewers. And so finally, I wanted to talk about our sequencing efforts. Before we've gotten started actually sequencing wastewater samples, we did a pretty large pilot using just SARS-CoV-2 genomic RNA that we had from the ATCC it's from the Washington state isolates. And so what we were looking at, we were comparing different RT enzymes, comparing different kits, and we wanted to look at both the gRNA itself, genomic RNA, and we combined it or spiked it in RNA extracted from 2014 wastewater, so shouldn't have its own SARS-CoV-2 signal at all. Part of the reason we did that is because we know how complex of a matrix wastewater can be, and indeed we were finding all throughout processing that there was a decrease in signal even when the same concentration of gRNA was spiked into the wastewater. However, we did get pretty good coverage and depth. So we settled on using the Swift Biosciences Amplicon sequencing kit, which should cover over 99% of the genome. And we were finding, here I'm just showing, the length of the SARS-CoV-2 genome. I put a little horizontal line at 2000x depth, but we see greater than 98% genome coverage. And also promising is that we expected 3 single nucleotide variants between the Washington isolate and then the Wuhan reference sequence. And so we were able to recover them with this sequencing method, this kit, and this depth that we were using. I should say we're using MySeq. And so then next we moved on to sequencing some of our samples that had the highest concentrations from our time series, just again, continuing to test out different samples and different methods. So here I'm just showing which of the samples we looked at, one from a peak in Dayton, one from a peak in Taylor Creek, and both of them were in the 10 to the 5th copies of N2 per liter range. And firstly, like I said, in our processing, we do RNA extractions from the filter fraction and the pellet fraction separately. So we wanted to process those separately for sequencing as well. And we found that the pellet fraction, we were having issues right from the cDNA synthesis step. We weren't really able to detect N2 by qPCR after doing that reverse transcription. And when we took it through still to sequencing, we had very few sequence reads, not a lot of mapping to the genome. So going forward, we're going to continue sequencing with only the filter fractions. With those, we did have really good genome coverage, so 98 to 99.78%. And we did this with a dilution series. So each of these 2 time points, the Dayton and the Taylor Creek, we diluted it up to a 1 to 8. So the lowest concentration was about 10 to the 4th gene copies per liter, and we were still seeing good coverage and depth at down to that level. In fact, we're seeing some improvement sometimes in the sequence quality when we diluted, which might be expected if there's significant inhibition issues or something like that. And so here I'm just showing, obviously the goal of all of this is going to be to identify mutations that are associated with the variants of concern that might be either already found in clinical cases in that community or might be emerging. So here, again, like I said, we're just looking at 2 samples, one from Taylor Creek and one from Dayton. But for each sample, we have 4 sort of replicates because we were diluting them at 4 different concentrations. And so I'm just showing some of the SNVs, the mutations that were detected across the genome. So going from 5' to 3', that's what each of those different x-axis labels are. We are seeing that, among the 4 replicates within each sample, we tend to identify mutations in all of those. I should say also the y-axis is the depth, right, for that base pair, how many, what the depth was in each sample. And we're already beginning to see that there are some mutations or variants that we're observing in the Taylor Creek sample that we didn't see in the Dayton sample. So this seems like it might be promising to start to identify emerging mutations. In this pilot, we did see the D614G, the G variant, which was basically at 100%, as might be expected. We have not detected any of the Nelly or other mutations associated with the UK variant, but I think that's also expected since these are from quite a few months ago now. And we're going to be moving on to assess current samples this week and onward. I should also say that we're hoping to find, oh, I apologize. I lost my train of thought. We're hoping, I should also say that a lot of the variants have a number of mutations associated with them, right? So for example, the UK variant has 13 different point mutations associated with it. So one of the things we need to think about in wastewater is we can only report, lists of mutations and whether or not they're co-occurring, but none of them, necessarily represent an individual. It represents an aggregate of the community. So that's just kind of an overview of our research. We know we're going to continue monitoring at least through June, probably beyond. And that way we're going to be able to, track what's going on throughout this process of vaccine deployment. This is just a screenshot of the vaccine dashboard as well. And then, like I said, there's an effort for developing Bayesian models that include fecal indicator abundances, flow, rainfall, recovery, and so on, to look at the relationship between clinical cases and the wastewater concentrations that we're measuring. And then finally, we have begun weekly sequencing to track the mutations that might be associated with existing and new variants of concern. So with that, thank you so much for taking the time to listen to this, and I will be happy to take some questions.
Thank you for that very informative and clear talk, Maitreyi. So I want to remind all the participants to please put their questions in the Q&A. So I'll start with the first question in the Q&A. So can you describe a little bit more of your main findings and recommendations on sampling at different spatial resolutions in the Mill versus Taylor Creek plants?
That's a great question. So I think that right now we are we've been collecting samples from both of those really regularly, and we're a bit behind. We're hoping to connect, fecal indicator measurements and flow and all those things. And we're behind on everything except for the actual the SARS-CoV-2 measurements. So I'm not sure that we have any recommendations yet, but hopefully we can work on that analysis.
Okay, thank you. We also have a question. Do you think that you can sort of definitively connect any of the SNPs to IDing the variants? Like, in other words, are some SNPs more predictive of certain variants?
So I know that in the preprints and the papers that have come out using sequencing in wastewater so far, the way that it's reported is kind of just reporting all of the SNPs associated with the variant and then providing percentages of samples where that SNP is observed. And I don't know how many I've seen where every single SNP associated with a variant are present. So I'm not sure that there has been a strong enough connection between the wastewater data and clinical data in the same sewer shed to really have an answer for that. But I know, like in Ohio, they're going to have us focus on, particularly on 3 different SNPs. One is the N501Y, one is E484K, and then one associated with the California variant. And then I think, so we're going to focus on looking at the proportions of those in each of the wastewater samples, and then there'll be ongoing work to look at some of the other mutations as well.
Okay, thanks. There are also a couple questions about some of the sequencing results, in particular about inhibition. So it sounded like you were having problems with the RT step with the pellets, and then you still had some inhibition from your filter extracts. And you mentioned that you had to do a 1:8 dilution. Is that because of inhibition in the filter extracts that you had to do that dilution?
So we suspect that it might be, but sorry if I was unclear. In the 2 samples of this pilot, we weren't seeing, like we were getting 98 to almost 100% coverage in all 4 of those samples. And it was just in one, I think it was the Dayton one, the 1:8 had even better coverage than undiluted. But I don't think that that's enough data to make any statements about whether dilution is helpful. I will say in our very preliminary results from all of our sites, which I just analyzed or started to analyze this past week, we did both undiluted and diluted for each of 12 sites. And we were seeing that in some cases, we got much better read mapping for the diluted sites. And in some cases, we were getting better read mapping for, or sorry, not diluted sites, but the diluted samples within a site. And in some cases, we were getting the opposite. And so I suspect that it's going to be a balance between the concentration at the site and being, whether it's too low for detection versus dealing with issues like inhibition. So for now, our plan is to do an undiluted and a diluted for every site that we look at, and then go from there as we collect more information.
Okay, thanks. I think we have time for maybe one more, maybe 2. So are you guys seeing an increased trend in the wastewater right now, given the rise in cases in Michigan, which is fairly close by?
I tried to peek at the latest stats, and I think that we are seeing, just from, last 2 weeks to now, we have been seeing an increasing trend at all of the sites that we look at. But I haven't looked at every single site in Ohio, so I can't speak to especially the ones closer to Michigan.
Okay, and I have one more question. So you said 50% of the population, 64 plants, and so that the widespread any state. Are the other 50% of the population mostly in rural areas or that use septic tanks? And so that's one part of the question. And then the second is, do you feel like the public health departments are finding the wastewater data is valuable based on your interactions with them?
So you're cutting out a little bit, but I think I got the question.
Or?
You are frozen, at least for me. I don't know about for everyone else, but I think I got it. And I do believe that it has been mostly urban populations that have been captured by the wastewater treatment plants. And the second part of the question was, could you remind me the second part of the question?
I can come on just in case Allie's email or internet is glitching out here. Just in relationship to the way public health departments are using the data, has your experience been that they're finding it valuable?
Oh yeah, so I'm not super plugged into the public health, component, but I'll say what I know, which is that I believe the standard was that if there's an increasing trend for 2 weeks in a row, then the local health officials are notified in that area, and then they can make decisions as they need. So that information is being deployed. It is also publicly available. And then with the sequencing effort, we're going to be definitely working even more closely with the epidemiologists who are looking at clinical sequencing data. And so now they want to look at sequencing data from those sewerships as well.
Great. Well, thank you. I think that's probably the end of time for questions. Thanks so much, Maitreyi, for your presentation. And we'll transition over to our second presenter, Devrim. You can go ahead and turn on your video and mic and share your screen when you're ready.
Um, do you hear me well and you see my slide well?
Yeah, you're great.
Okay, cool. So hello everyone, and thanks for joining us for today's webinar. And I'm Devrim Kaya, I'm from Oregon State University, and today I'm gonna talk about our efforts at Oregon State, and my title for this talk is I will evaluate the SARS-CoV-2 prevalence and dynamics from building to city scale by wastewater-based epidemiology. And this slide shows the summary of our efforts. And we have 4 projects going on, and they are supported by different foundations. The first one is NSF Rapid. Under this project, we do micro-sewerage surveillance in collaborations with the Clean Water Services. And the second one is internally funded project. We call it Trace OSU, and we monitor SARS-CoV-2 at the building dorm level, at the campus level for all 3 OSU campuses. And the third one, it's the Trace community. It's founded by David Lucille Packard Foundation. In this one, we combine WBE with random house-to-house nasal swab sampling to determine the true COVID-19 prevalence in a community. And the fourth one, we collaborate with the OHA, Oregon Health Authorities, and CDC, and it's a statewide monitoring for the virus, COVID-19 disease, and the genotypes at 40 plus wastewater treatment plant across the Oregon state on a weekly basis. I'll give you a brief introduction for our methods. And we collect samples from wastewater treatment plants influence or from manholes. And we filter it, and to concentrate the virus, we use electronegative membrane, and we homogenize it with bead beating, and then we use an automated RNA purification system. It's a Kingfisher, and we use droplet digital RT-PCR, and our targets are M1 and 2 for SARS-CoV-2 detection and quantification, and RP as the human RNase P gene as an internal control, and BCOW as the process control for recovery. And we report to the authorities. And first, I would like to give you some method-related information. Under NSF project. So we did first, when we started, we were wondering if we should get grab or composite, and what would be the differences between those sampling strategies. So in this figure here, let me use a laser pointer. So in this one, we have the SARS-CoV-2 concentration at the y-axis and the hours we did sampling. And at the wastewater treatment plant, the orange line shows the composite sampling value, while the dots and the line here, the fluctuating line, shows the grab samplings for the SARS-CoV-2 concentration. As you can see, it fluctuates, but it's overall close to composite value. Still, if you do, sometimes you can get a lower signal. When you look at the micro-sewerage neighborhood flow, and you see the variability is higher compared to the treatment plan, and it varies throughout the day because of the people's usage of the water can change based on their habits. And the wastewater treatment plant equalizes the flow fluctuations. And when we go even smaller scales, like city block level, you see if you can miss the variability even higher, and at certain times you could miss the signal. And lastly, the single building flow, it is quite even higher, and the chances to miss the signal is higher, the highest actually. So from that experiment, so we have been using the autosampler 24-hour composite sampling strategies. And now the question is, can the wastewater-based epidemiology identify the presence of COVID-19 infected individual in a building? So look at the building scale. So Under Trace OSU project at the building level, we monitor our campus, OSU campus, and we have 23 sampling sites. And this circle shows our samplings, which covers most every residence halls and most Greek houses. And we do twice a week sampling, and we turn the data around rapidly, and we report to OSU COVID response team. And as I said, we do 24-hour composite sampling, and we have auto samplers to do that. And the OSU COVID response team used this information to determine where to do additional nasal swab testing to identify the infected students. So I'm going to show you a case here. So this figure shows the SARS-CoV-2 concentration variation in the time series of this virus in the wastewater we got from one dormitory. At the beginning of, at the mid or end of the October, we see a spike in the wastewater. And then our OSU COVID response team initiated the testing right away, and they were able to identify one infected student. And then later on, at the beginning of this year, we saw another high peak in the wastewater for SARS-CoV-2 concentration. At this time, they didn't do testing right away, so the signal kept increasing. And later, like in a week, they did the testing and they identified the 3 students. So when we identify the infected students, they are given two choices. Either they can stay in the dormitory, in their room and isolate, or they could go to their homes and isolate there. In this first case, the student decided to go home. As you see, it was reflected by the wastewater SARS-CoV-2 concentration. But in the second case, these students decided to stay in their room and isolate. As the time went by, so they recovered, and we were able to still continue to see the signal in the wastewater, but it was decreasing. So benefits of the building level scale and monitoring is you can find unknown infected individuals, and you can use it. It's a good safety net in a campus surveillance project. So the authorities can initiate the measures and take the precautions before it gets spreaded a lot. And there are challenges as well. So the severe surveillance can pick up the transient individual. So maybe a student's having a guest and coming and using the restroom and out. So you may not be able to identify the student. Or individuals may choose not to get tested. And also, as we know, individuals may shed the virus for 2 to 4 weeks after infected students returning from the quarantine as well. And autosamplers failures and clogging happens a lot. I'm sure most of you have experienced that. So there are a lot of challenges with those monitorings as well. So at the community level, the question was, can wastewater-based epidemiology identify the hotspots of this disease within a community? So this is a Newport civil shed study. It was our first study for the community, and it involved nasal swabs as well. So in this map, you see 22 different neighborhoods. And so this happened early June 2020. There was an outbreak, and our team went there and did sampling 2 weekends in June and July, 3 weeks apart. And in conjunction with the trace nasal swab sampling was done. It was door-to-door nasal swab testing. And yeah, we look at the 22 pump station plus the influent of the wastewater located down here. And all these wastewaters get together and comes to the wastewater treatment plan. So the spatial analysis of this wastewater and the sampling and nasal swabs data is going on, but I'll give you a little information here. So here in this first figure on the left side, it is from June, and the color coded shows the wastewater SARS-CoV-2 concentration. As the color gets lighter, the virus concentration is increasing. As you see, we were able to identify the hot spots in June. For example, the yellows are the highest SARS-CoV-2 concentration areas. And also these red dots shows the clinical OHA reported cases. And they were correlated together, correlating well as they were clustered in the hot spots. And 3 weeks later, we did another sampling event in July, and we saw there was a decrease in the SARS-CoV-2 concentration in the wastewater, and there were fewer or less cases, but again, associated with the high concentrated virus areas. And in these 3 weeks, there was a mask mandation. That's why it went down. And our another efforts to show this is consistently, we can find hotspots within other communities. So we did overall 10 sampling events, several weekends for some of them. And at 7 communities, we did nasal swabs incorporated with the sewage shed sampling events. For example, at Newport, it was 22. And in Core Valleys, we are located, the main campus is in the Core Valleys. We had 8 locations. In the Boardman, we had 3 locations, Hermiston, 4 locations. And yeah, so many locations. In Eugene and Spring, we had 17 locations. So when we look at the data for the sewer sheds, we were again able to see hotspot areas, like the red is the highest concentration for the virus in the wastewater, and there were green areas with moderate or very low at the detection level for the virus. So this is for Band, and then we have Redmond as well. And we were able to locate a safe, kind of safe and hotspot areas. And I'm not going to show all of them, but as you see, some cases there were all over, the virus was very high, and it was reflected with the nasal swab as well. As in Hermiston, the prevalence was 17%, while in Newport here, it was 3%, and then it reduced below 1%. And then another question at the statewide, can we look at track the dynamics of the disease throughout the Oregon state? So for this statewide surveillance, we have 40 plus wastewater treatment plants, and we do weekly monitoring started early September, and now we are increasing to twice a week. And the project duration is 2.5 years. And we report to OHA here in this case, and Oregon Health Authorities, the dashboard here, they are just showing detected or not detected by the cities we are following, and adjusting their policies and the vaccine campaigns. And when we look at the fluctuations in each city, we were able to detect the low cases, high cases. These are all wastewater SARS-CoV-2 concentration and the time series beginning of September. And so this was very well correlating with the COVID-19 cases, clinical cases. And from here, we started with less than 16 counties, and we kept increasing. Right now, we have 24 counties. There are these gray areas. Those we haven't gotten participate, but there are rarely the population is very low in those areas. It is very terrestrial areas. And from October to this month, you can see as the color change shows the virus increase, and we are seeing increase in the wastewater still. It is at 10 to the 5th gene copies per liter, or sometimes even higher. So yeah, we have expanded to include at least one city in each county. We have generated thousands of data points from about 3,000 samples, and over 1,400 samples we have sequenced. So now let's look at a little bit preliminary data for the sequences. This shows the abundance of these variants in our wastewater. These for sure not only variants we see in our wastewater, but the others, below, they are below like 0.1%. So we aren't showing those. So the orange one shows the variant we call FF previously, but now it's called B12. And it has the dominant one going on in Oregon State. Then we started to see increase in the Californian variants beginning in November. But we analyze only positive samples, and around log 4 samples for the waste. When we get a log 4 or above, then we sequence all samples for looking at the mutations and the emerging variants and the variants of concern. So this UK variant, for example, it is have greater risk of mortality, and we started to see a increase in these variants. And the Californian variant, it was first emerged in California in summer of 2020, and it is associated with the high increase of person-to-person transmission, and now it is spread to 25 other countries. And our wastewater sequences results are accorded with the sequencing of the clinical samples from the same regions. And this is the OHA dashboard. They are reporting the variants of concern, how many were detected in which regions, and our results are in line with the clinical data. And can we estimate the COVID-19 prevalence? So let's look at, remember this figure, we went to all these towns, 10 events several times during the weekends, like 2 events in a weekend, and 7 towns. So we surveyed these towns for nasal swabs as well as for their neighborhoods data for the virus concentration. And when we put together this wastewater data, and correlated with the reported clinical cases, and the correlation was around 0.5 for R square, and this is the y-axis shows log reported cases per 10,000 population, so they are normalized to the populations. And these are the influence concentrations of those cities. 7 towns only, not all Oregon, by the way. So the R square is comparably low when we look at when we compare the SARS-CoV-2 concentration coming in the wastewater with the true prevalence. The true prevalence here the data coming from the nasal swabs. And so the correlation, when we put together all those towns and the dates are here, the R square was 0.91. So it is very highly correlated with the wastewater data. And we can say wastewater data can be trusted and unbiased, because those cases, as in this case, the reported clinical cases are highly biased, as you may also observe in your states, but you cannot hide from nasal swabs or the wastewater capture asymptomatic cases as well. So yeah, this was quite good correlation. And each data point is very expensive to get these studies to do, and $200,000 spent to get one data point. So it's quite a large effort and expensive. And the lessons learned, and so we were able to detect a single infection at a building scales, and high resolution spatial sampling, we were able to pinpoint the infection hotspots. And the wastewater correlates more strongly with prevalence than the reported cases. There are challenges and opportunities, depends how you look at those. We need to still lower the sensitivity, because as the pandemic evolves to the endemic, we may need lower sensitivities to detect the disease or to track the disease. And we need to still shorten the turnaround times, and we need to standardize the methods and result interpretation between the labs, and continue optimizing the communication of results with the public, because it is hard. We report to, in this micro sewerage neighborhood, we report to the county mayor, and yes, so there is a large team behind of these efforts. There is a wastewater team, and PI are Taylor Rednicki and Christine Kelly. There is Michael, he collects and coordinates students, numerous city employees. I would like to thank them all. We have sequencing analysis team, Brett Taylor, and his postdoc Dana and Katie and Mark in the analysis team. And we have a state lab from Clean Water Services, and Ken Williams and Blight Layton we are collaborating together. And the OSU Student Health Services team, so they collect the nasal swabs, and we have statisticians, epidemiologists, and there is a huge efforts and support, and I would like to thank them all, and thank you for listening as well.
Thanks so much, Devrim. That was a great presentation. We have time for some questions. So if anyone has any other questions, just a reminder to please put them into the Q&A. I'll go ahead and start off. So first question is about your variance data. So can you clarify how you are detecting variance in your wastewater samples? Are you using sequencing or qPCR assays?
So we do sequencing, we do high-seq sequencing. My-seq wasn't covering good, the coverage wasn't that good. So we use a Swift kit, and when we get a positive data from our samples, and when the log is above 4, we send them all for sequencing. For log 3, and we have also started to do those as well.
Okay, thanks. And then just a quick follow-up. I might have missed it when you were explaining. So for that variant data, then I think your plot had something like fraction of wastewater RNA with variance. Is that then a number of samples that had that variant in it, or is it a quantification based on the sequencing data?
It is like abundance, and it was normalized to the statewide. And I can show you again if, let me see, share that one. So I think you are talking about this, right?
Yeah.
And yeah, so those are abundance and statewide weekly fraction in the wastewater. So it is kind of normalized to how many samples we get, and based on RNA. And if we see a sequence, and the sequence, the reads, the number of the sequence we see in a sample, and statewide. So it is normalized like that. This is the abundance. So we have other variances going on, but they are very low at level, so we didn't put them here. So
okay, so each point is essentially potentially from multiple samples around the state, and you're normalizing against the number of hits you're getting on.
Yes,
on
exactly.
Variants. Okay, thank you. I think we'll move to the next question here. Was more related to some of the work you presented at the beginning about grab samples versus composites. So in looking at that, I know that a lot of places are having trouble, especially in lower middle income countries, getting auto samplers. And so obviously your data shows that auto samplers and composite sampling is certainly preferable. But in analyzing that, do you have a strategy that you might suggest for making grab samples something that you can still get quality data out of?
I think so. When we look at this figure again, and so these are the points for grab sample points. So if they go like between these hours, so they would miss the signal. I would suggest they should do at least 3 times a week. First getting the grab samples and see which, to decide what is the habits of this town when taking samples. And they should look at their signal and their signal fluctuation during a day, and at least 3 times I would suggest to find, oh, maybe this town, because it may change if it's then into the very high, how to say, industrial area. Maybe people there would wake up early and use the restroom earlier than a touristic area. So they should be applying in different strategies, and they should sample their sewage sheds based on their strategies. Yeah, because one strategy may not apply to all places.
Yeah, and it seems from your data as well that obviously the population contributing is important for the variability, like you mentioned. So grab samples of a larger population, maybe not as much of an issue.
Exactly.
You mentioned that. So
yeah,
definitely location specific. So again, about your on-campus data, how is that shared with students? Is there a public dashboard, or do you know if there's a notification system for the wastewater data?
Yeah, so we do contact trace, as well as we report random, the number of the cases at the campus. So we have a dashboard, and we have a dashboard for wastewaters, and the student health services also have those as well. So we report if students or faculty, we don't, of course, report the names, but the numbers of the cases are reported.
And maybe also along the same line, there was a question about building level data, and sort of you mentioned a similar challenge here about distinguishing between post-infection fecal shedding from infectious state shedding. And I'm curious, with the levels of different data that you've collected, especially in times when you're able to pinpoint cases leading to that shedding, have you noticed any sort of impact of that on the data, or some way to try to account for that when you know that might be occurring in building level data?
Yeah, since we regularly follow the, get the samples and look at the virus concentration in the wastewater, so we were able to see when it was going down, when it started. So we were really able to pinpoint those cases. As you may remember, at the beginning, it was like stable, zero, non-detect, and then we could see a peak, and then the testing would be initiated. And yeah, I think it is very correlated, but we still need to do some more analysis with the data, and to be able to maybe report based on the students, the shedding rate and the cases.
Great, thanks. And I know we're close on time, but we have one final question that I think I'll try to fit in here. So again, going back to your sequencing data, if you can just really quickly touch on how you're calling your variants. And so kind of, we had 2 presentations with a bit of sequencing data. Is it all mutations on that variant that are being called to designate that a variant was detected, or is it specific mutations?
So I don't do that part. So Brett Taylor and his team does it. Okay. But so our wastewater sequencing, since we also do nasal swabs at the campus for the students, so they were really correlating really well. And those mutations, I think they are 99 or over % identity to the reported, the UK variants or the Californian variants. So we have seen, I'm not sure if I'm answering your question right, or if this is the answer for
Maybe
your
we
question.
can, yeah, we're close on time, so maybe I'll
Yeah.
have, that was from Nicole at EPA. You guys can get
Okay.
in touch
Yes. Yes.
afterwards if
Please.
needed to
Yeah. Contact
clarify.
us.
Yeah. Well, great. We're right on time. So thank you so much to both of our presenters, Devrim and Maitreyi, for joining us and sharing your work today. And thanks for everyone for attending. We'll have another webinar coming up in 2 weeks. So we hope to see you then.